Actually, I give you 4 minutes for that. Anyway, you guys, I highly recommend watching that video. Since everything you're about to learn, it's condensed. It does it a lot quicker. Let me see here. Before I take your questions, you pull something else up. Watch those later. I feel like the go too much into detail. Those are for chemistry classes. We're going to talk a little bit of chemistry today again. All right, about that later. Okay. Do some attendance and answer some questions. Bryant is here in WPSU gear. Curry. Garvey. Fisher. Hamilton is here in WPSU gear. Melman. Ashley's here in WPSU gear. Crystal is here. Taylor's here. Norris. Rollins. Saunders. Saunders is here. Champlin is here and WPSU here. Nope, never mind. Wrong school. Neither ranked in college football, but you know, pick your one. Talking this trash. Taylor Pierce, Vargas, and Young. I didn't call. And we still have about 2 minutes, but we are recording. So to answer your questions, the exam is now reopened. So that initial invite you got, you can just use that. If you can't find the exam, send me an email and I'll send it to you again. Curry is here. Anyway, so for the retake, you can take it as many times as you want. And it's open book, open notes. It's not timed, right? So just take it, like I said, as many times as you want. And then the only thing you need to do is let me know when you're done. Because what I'm going to do is take your first grade, take your last grade, and then average the two. Make sense? And then even after that, you'll still have access to it because you're going to need to use that to study for your final. But yeah, so that's the situation. Yes.
So we don't have to meet your office hour or anything.
Correct. Yeah, you don't have to meet me. It's completely on your own. It's open. You have availability to it. The only thing I would ask you to not do, you can even work together, like, but not copy, if that makes sense. Like if she's gonna do it and then you wanna help her, sure. And then if she's gonna do it, you're gonna help her, sure, but you can copy off each other, if that makes sense. I have no way of knowing if you do or not, but that's not gonna help you learn anything. So any questions? If for some weird reason you were to retake the exam and do worse, then just don't tell me about it and we'll just keep your original higher grades. I don't know how you would do worse, because like I said, it's overflowing and all that. And the deadline will be anytime before the second exam, right? As long as you get it done anytime before the second exam. The only reason there's even a deadline is because once we pass the deadline, then I can meet any one of you or all of you and give you the answers. Like we talk about your exam, like we'll look at your exam, talk about why your answers are wrong. You know, I can give you feedback. So not only that, but you could also get a 5% boost to your grade for meeting me online and let me give you feedback on your exam.
So when is the projection for the second exam? And will it be like 108 or what was this last time?
It's looking like it's going to be like it was last time. And I would say probably we'll finish this chapter this week, hopefully, and then we'll finish the photosynthesis chapter next week. So hopefully it'll be the week after that.
So the first week of October.
Sounds right. Yeah. Any other questions? Well, it's 8 o'clock. Let's get back to attendance. Let's see here. Young's online. Fisher was online. Harvey? Here at WPSU here?
Yeah.
Correct school. Hellman's not here, correct? Horace is here.
It's under this, you know, it takes us off.
Sounds weird. Yeah, I'll just trust you. Be offering you extra credit to take your clothes off. Macy Rollins, Taylor Pierce, Vargas. Everybody else is accounted for, correct? All right. Hey, what a mess. I don't know if you guys saw my announcements. The wrong PowerPoint was uploaded to my NPSU. But I didn't want to stop what I was doing to look into it. So it wasn't a complete waste on Wednesday. You did learn some stuff that you do need to know. Only problem is now I need to kind of go back. So what we're going to look at is kind of backwards. I won't say backwards, but we're backing up. Like if we were going through the chapter, I kind of need to now go back here, right here into the chapter. There's some stuff in the textbook that was not on that PowerPoint slide presentation, but I've got the correct one now. So let's look at it. And this is important because had I not noticed that and you read the textbook, you would have probably been confused because you would have seen this stuff and they're like, what in the world is this? Right? You never mentioned any of that. So, for example, this stuff, I'm not going to ask you about. There are some questions about it on the study guide, but I'm not going to ask you about this on the exam. Long story short, like we said before, cells need energy to live, right? And your book is pretty specific about it. If you've had a chemistry class, then you're kind of familiar with this. So in chemistry classes, real chemistry classes, when you're doing chemistry, you're using equations, right? And everything's quantified, everything's mathematical. And that's all this is. So if you remember when I was talking about entropy on Wednesday, I said you don't need to know this, but it's in your textbook. And entropy is important for chemistry when we're doing actual chemistry or quantifying things using math. That needs to be basically represented mathematically. That's what entropy is for as far as we're concerned. Same thing here. Free energy, well, abbreviated as G. Again, you don't need to know that. It just quantifies the energy that's available for doing work after accounting for losses. So again, not that you need to know this because we're never going to use it again for the rest of the semester, but just so you know, at least for the study guides, if delta G is negative, so delta G means a change in energy. If the change in energy is negative, right, which you'll never see a negative, if I'm never going to give you a chemical equation, that just means energy is released. And if it's positive, that means energy is absorbed. If anything, and not for the exam, but maybe we'll use these words again, and I really doubt it. Exergonic reaction and endogenic. And it's kind of in the name, right? Exerg, like giving out, going out, right? Energy is released. Endergonic, energy is. So if anything, you might need to know those words because maybe we're interested in it, but probably not. So any questions about all this stuff? I know we have one more bullet point. Are there any questions about free energy or exergonic reactions or endergonic reactions? All right. So whether it's exergonic or endergonic, right, whether it's releasing energy or absorbing energy, all reactions require an initial input of energy to get started, right? So even like, for example, a nuclear explosion is a very exergonic reaction, right? It's a lot of energy being released. But even though you're releasing a lot of energy, you still need to have a little bit of input to start that reaction, right? So that's what we call activation energy, and that is important. That'll be the first word too for attendance, energy. So if you're watching the video, not if you're online right now, but if you're watching the video later, you take a screenshot of me pointing at the word energy. Anyway, yes, all reactions require activation energy, and we're going to talk a little bit more about that in the next slide. So far, are there any questions? All right, your book defines activation energy as the energy invested to start a reaction. Here, I'm going to go back to this analogy that I've been using. Remember the crossbow, right? It said however much energy it takes to build something, that's how much energy is ready to be released. Same with a molecule, right? However much energy it took to build that molecule, then energy will be released when you break that molecule. Same here. However much energy we put into that crossbow is ready to be released. And it's a lot of energy, right? That you can kill somebody with that thing. But it doesn't shoot itself, right? How do you get the crossbow to release the energy? I mean, it's in the picture. How do you get the crossbow to release the energy that you put into it? Pull the trigger, right? So that would be the activation energy. The energy required to pull the trigger, right? That's your activation energy. Yes, there's a lot of energy ready to be released, but you have to pull the trigger. You have to have that activation energy. We're going to talk about enzymes later. This is what I'm getting at here. So with enzymes, as you'll see later, basically all they do is lower the activation energy. So I don't know if you guys have ever shot guns before or crossbows, but you know, some triggers are a lot easier to shoot than others, right? For those of you who have. So you can think of an enzyme as switching the trigger from something that's like really hard to pull to something that's like really easy to, you know, you barely touch it and it fires off. Because again, you're lowering the activation energy. But here's what it actually looks like. This is the picture from your old textbook. So here we have, this is how much energy is stored, right? Remember, chemical energy is a form of potential energy. This is how much energy is stored in that molecule. It's a really high energy molecule. And if we break those bonds, we release that energy, right? So the products have a lower amount of energy. That energy is being released. But it can't just happen without some sort of input of energy. And that is what activation energy is, right? Again, you need to get over that hump to get it down. And an enzyme, as we're about to talk about in the next few slides, and then an enzyme lowers the amount of activation energy that is required. So that's the picture from your last textbook. Here's the picture from your actual textbook. Same thing, right? We're just showing, in this case, we have actually more energy in the products. But either way, the point is we have activation energy required to get to where we're going. So I like the other picture better, but I wanted to include this one in your textbook so you can follow along. Anyway, any questions so far about activation? Remember, it's that energy needed. Another good example would be, I don't know, if you were standing on top of a mountain and had a bowling ball, right? Obviously, that thing's going to roll down the mountain, but you give it a little kick to it first, right? Or push. I don't know why you would do that, but that's a good example of you need that little extra energy just to get things started. And once you do, then they can usually take care of themselves. Some things you need to know for the exam and for the study guides. One is this: enzymes are very selective. They're very specific, however you want to say it. Like usually there's one enzyme that does one chemical reaction. So enzymes are involved in chemical reactions, again, because they lower the activation energy. Looking at the next slide. Yeah, so as a matter of fact, and I don't know if I said this before, so I'll make sure I say it right now, even if I'm repeating myself. A lot of the chemical reactions, almost every chemical reaction in your body, will not happen without enzymes. Just not enough energy, activation energy, or too much activation energy is needed, so they just wouldn't happen. So without enzymes, we wouldn't be alive. But yeah, that's the first thing you need to know about them. They're very selective. Let's talk about some words you definitely need to know for the exam, and because I'll be using it later in the semester. Enzymes recognize something called a substrate. You need to know what a substrate is. That's the reactant molecule. So whatever the enzyme is doing its job on, that's the substrate. So in this case, the substrate is like a little curvy thing in a triangle thing. They don't give it a name in that example. That's what the substrate is. Now, the substrate attaches to the enzyme, or the enzyme attaches to the substrate, however you want to say it, at the active site. So you need to know that too. As far as I'm concerned, that's probably all you need to know about the active site, right? That's where the business happens, just say that. But yeah, your book does get into, actually your book really goes into detail about how the chemistry fits too. Obviously the shape fits, you can see it in this picture, but the chemistry fits too. So for example, if this was positive, that was a positive part of that molecule, then obviously we would have a negative part of a molecule right here, right? Because those things go together. Or if this was hydrophobic, then this would also be hydrophobic, right? Because hydrophobic and hydrophilic things repel each other. Just an example. But your book goes into a lot of detail about side chains and amino acids. And you can just skip through that. I mean, read it, but I'm not going to ask you about that. The point is, the active site is where the business happens on an enzyme. And it's because the substrate fits there physically, right, with the shape and also chemically. Anyway, when this happens, then the enzyme usually changes shape. And this is what's called an induced fit. And you need to know that too. Induced fit is the entry of the substrate, or once the entry of the substrate happens, it induces the enzyme to change its shape and fit between the substrate and active site stuff. And I use this picture as an example. We all know what a hug is. We all know how to hug. Like a hug ends up like we can see in that picture, right? A hug ends up like this, correct? But it doesn't start that way, right? Imagine if I was trying to hug somebody and I walked up to them like this. That wouldn't work. Or there might be some cultures where they do that. I don't know. Likewise, though, if I were to do this, because right, this is how you start a hug. If I were to just do that and keep it like that, that's kind of weird too. Right? So it's more like an induced fit. You come in for the hug, and then once you're there, then you bring your arms around. That's sort of what happens with the enzyme. The induced fit. That substrate attaches, and then the enzyme kind of snugs itself around it. Which makes sense, especially in this picture, right? So in this picture, we're doing catabolism, right? Because remember, catabolism is the type of metabolism that breaks something down instead of building something, right? So in this case, I know it's hard to see, but imagine like there's some chemical bonds right there, right? And the enzymes can physically like put strain on those bonds, just like you could physically grab something and like bend it. That's exactly what's happening. It puts strain on those bonds. And of course, it also puts strains on the bonds chemically as well. Anyway, any questions about this slide? All right, the next word for attendance then, since we're talking about it, that's hug. H-U-G. Like what those people are doing. Hugging. We're going to talk a little bit more about enzymes, but are there any questions so far? In your last exam, or excuse me, the other textbook we used to use, this was in the previous chapter. So don't be for a loop. Anyway, this is also another important thing to know about enzymes. After the product is released, the enzyme can then accept another substrate molecule. You don't necessarily need to memorize that verbatim. There's all kinds of different ways I can say that. In other words, enzymes are not used up in a chemical reaction. And I'll give you an example because I might have a test question like this. I don't know yet. If I do, it'll be rare because it's not on the study guide. I might give you some chemical reaction you've never seen, where I have like, I don't know, A plus B gives you B plus C. And I'll be like, all right, which one of those is the enzyme? Of course, I'll just use actual enzymes and actual molecules. But the answer, the way you could tell, answer is B. B is the enzyme. And the way you can tell is because it wasn't used up in the reaction. A was changed into C, whatever that is. The B is still B. So that's how you can tell that an enzyme is an enzyme in a chemical reaction. I probably won't ask you that. Now that I'm thinking about it, I guess if I do, maybe it'll be extra credit. Because again, it's not under study guide. But you do need to know that an enzyme is not used up in a chemical reaction. It's not changed. It's exactly the way it was. So any questions about those two bullet points? All right, something else that's worth noting, and I don't know if I'm going to ask you on the exam or not. I think there are questions on the study guide, but enzymes, just like any proteins that we mentioned when we first started talking about proteins, they are sensitive to their environment. So when things heat up on the molecular level, what does that mean? When I say it's getting hot, I've said this a lot, so hopefully somebody can do this right away. When I'm saying things are getting warmer, what does that mean at the molecular level? What's happening to the molecules? They're moving faster, right? Yes. So for that reason, generally speaking, as things warm up, reactions start happening fast because the molecules move fast. Literally, they need to hit each other, right? This thing needs to hit this thing. They're just floating around. So the faster they float around, the more likely it is they'll hit each other. So yes, you do need to know that at the very least for the study guide. Generally speaking, the warmer they go, the faster. However, every enzyme has an optimal range. So too hot, we'll talk about what happens when it's too hot later. But if it's too cold, it just, it doesn't work right. So every enzyme has an optimum range. As you might imagine, most enzymes for humans, their optimum range is right around 98.6 degrees Fahrenheit, right? Because that's what we are. But there's other enzymes for other organisms that are completely different. We don't really have time to talk about it now. We might talk about it later. What will happen though, if it gets too hot, is it'll denature. Because remember, it's chemical bonds holding those things together. Remember, proteins are polypeptides until they fold and get that correct shape. There's all kinds of different bonds holding them together. Sometimes there's ionic bonds, sometimes there's covalent bonds, sometimes there's really weak hydrogen bonds. And as you add heat, you're adding energy, and then those bonds basically break. And when that happens, basically the enzyme, which is the protein, unravels. But the proper word for that is denature. So know those three things at the very least for the study guide. It's also worth noting that pH mess things up. We already talked about that, right? When we first started talking about proteins, I talked about pH because if things are too acidic, right, you have a lot of H+ ions floating around. So obviously, if there's any kind of ionic bonds or hydrogen bonds in that protein, then that's going to disrupt it, right? Because it's going to be attracted, that H+ is going to be attracted to negative. Likewise, if things are too basic, you have a lot of OH-, right? But again, that will disrupt the hydrogen bonds or the ion bond. That's a negative thing. It's going to be attracted to the positive things. And then also, this is worth mentioning. I won't talk too much about it, but it's also affected by things like salt concentration and coenzymes. But we'll talk about the coenzymes a little bit later. And then finally, the last thing you should know. Again, I don't think I'm going to ask you this because it's not in your book. It was in your last book, but I think it's a good thing to know. They usually end with ACE. So enzymes are usually named for what they do, and they end with ACE. Right? So, for example, on this one over here, this is a real enzyme. It's called lactase. You can tell it's an enzyme. Well, one, because it says enzyme, but it ends with an ACE. And guess what its substrate is? Based on the name, what do you think its substrate is? What kind of thing do you think it works on? What molecule? So, because remember, they're named after either their substrate or what they do. So, any guesses of what molecule this thing works on?
Lactose.
Good, lactose. Yes. So in this case, there's your substrate, it's lactose. Then the purpose of this enzyme is to break this down, break down lactose to galactose and glucose. We have a hydrolysis reaction. There we go, galactose and glucose. And then the products are released, and again, the enzyme is ready to do its thing again. So again, I guess I might have a question on the exam, like I said earlier, where I give you an equation you've never seen before. And there'll be a question that says, what is the enzyme? And again, the way you can know what the enzyme is, it'll be on both sides of the chemical equation, and then it'll end with ACE, right? And then I'll say, what is the substrate? And remember, the enzyme is named for the substrate, so just look at the things on the chemical reactions that look like they're named after the enzyme, or vice versa. And again, I guess if I do that, it'll probably be extra credit, because it's not in your textbook, it's not in your studies. Anyway, any questions about this slide? All right. Is anybody here old enough to remember fidget spinners? Yeah. You guys remember they were like a big craze, like for about a year, and that was it. They just kind of went away. But they were a big craze. So imagine if you were a factory owner. It's the fidget spinner craze. Like you just pour all your resources into that, right? Your employees, your machinery, your materials, right? All your time and effort just building fidget spinners because now's the time to do it, right? But then once they stopped being so popular, you needed to adjust things, right? You don't need to make so many more. You make a little bit. But now you need to adjust to make some other toys. that's sort of what enzymes are. And we have these things called enzyme inhibitors because these enzymes in your body or any organism's cells, they're making product, right? Enzymes are making things. They're breaking molecules down, making ones, and they're making stuff. But your cells don't always need the things they're making, so they have to be controlled. Enzymes need to be like turned on and turned off. And that's where we get enzyme inhibitors. You need to know what they do. And it's in the name. Enzyme inhibitors inhibit metabolic reactions, right? They keep the reactions from happening. I think usually people, when you think of the word inhibit outside of biology, I think of the word I hear the most, but the usage I hear the most would be like, they lost their inhibitions, right? If someone drink too much, they lost their inhibitions, meaning they lost the thing keeping them from doing stupid things. So again, inhibitions, inhibit, right? You stop something. So you need to know that about enzyme inhibitors. They inhibit metabolic reactions. Now you don't necessarily need to remember why, because I was telling you the reason why is because your cells don't necessarily need whatever that enzyme is producing. And at those times, the enzyme needs to be turned off. That's just for your knowledge. But you do need to know they basically stop those reactions from happening. How does it work? Well, there's different enzyme inhibitors, but they all bind to an enzyme. So they physically attach themselves to the enzyme. Enzyme inhibitors bind themselves, they attach themselves to the enzyme. And it is that binding to the enzyme that actually does the job. It's the thing that disrupts the function. So any questions about this bullet point? You need to know this, all that stuff. All right, so there's two different types of enzyme inhibitors that we're going to talk about. The first one is competitive inhibition. And it's really simple. It simply plugs up the active site. So this is a little cartoon I found. I didn't make it. But it's a perfect illustration of competitive inhibition because, so here's the enzyme right here. This is the actual substrate, right? You can see it's got the shape that would fit in there. But here you have this competitive inhibitor. And all it does is it goes in there and it plugs up the active site. So now, because that thing is there, the actual substrate can't come in, the enzyme can't do its job, and it's disrupting the function. Does that make sense? Competitive inhibition. And there's one more that we're going to talk about on the next slide. Just out of guess, let's see if anybody can figure this out. If we're talking about competitive inhibition, does anybody want to guess what the next one's called? I don't expect anyone to know, but I'll see if anybody can guess it. If this was called competitive inhibition, there's only one other category. What do you think it might be? And I guess that's okay. Non-competitive inhibition. So again, it's still binding to the enzyme, but it's not binding at the active site. It's binding somewhere else. And that's basically all you need to know. Well, now, I guess that's not good enough. Let's also talk about this. Once it binds to the enzyme, not the active site, then it changes the shape of the enzyme. So it's not that the active site is blocked anymore. It's just that the active site has changed shape. So now the substrate can't get in it. And this is also known as an allosteric inhibition. As far as we're concerned in this class, these two things are the same thing. Non-competitive inhibition and allosteric inhibition are basically the same thing. You can look it up for independent work and look into the differences, the relationship between those two. Again, they're basically the same thing, but if you get really specific and really dive into it, technically there's a difference. Anyway, another thing that's not on this slide, speaking of that word allosteric. So like I said, for competitive inhibition, which we talked about on the last slide, right? It plugs into the active site, right? For non-competitive, and it's not on the slide, it plugs into the allosteric site. This is in your textbook. Hence the name allosteric inhibition, right? So in this case, the inhibitor binds to the allosteric site, which again should be a little point. It's not on there. I need to fix that. Any questions about this before I show you kind of a picture of both the two different types of inhibition? All right, this is a picture from your last textbook, so they don't have the proper words. I just don't even give them the names. But yes, so here's the one type, right? This is competitive inhibition. Well, no, sorry, this is just an enzyme working, doing its thing normally. There's the enzyme, there's the active site, there's the substrate. The substrate fits, it does its job. Here we have competitive inhibition, right? So in this case, we have this inhibitor that blocks the active site. So then the substrate can't get into the active site, so it doesn't do its job. And then we have non-competitive, right? And again, this picture is not from your textbook. Because if it was, this would say allosteric site, right? So the inhibitor moves there to the allosteric site, which changes the shape of the enzyme. So again, the active site's not clogged, it's just a different shape. So now the substrate can't fit, and again, it doesn't do its job. Make sense? All right, there's even more about enzymes we're going to talk about. So obviously, we've talked about how enzymes need to be turned off, right? But also, of the same note, they kind of also need to be turned on. There needs to be on and off switches, which is what this slide's a little bit about. Enzymes often require helper molecules that can form temporary or permanent bonds. I would say the most important thing in that bullet point is just this. Enzymes often require helper molecules. Yes, they can form temporary or permanent bonds. The important thing here is you need to understand that sometimes enzymes need helper molecules. And the two that we talk about are cofactors and coenzymes. Now, I do mention a coenzyme later in the semester, actually even later in this chapter. But as far as I'm concerned, I don't think this is very important either. It's not so important that we're going to completely skip it, but I don't think I'm going to ask any questions about it on the exam. There are questions about it on the study guide. So basically, the difference is cofactors have inorganic ions. The book examples were iron and magnesium, right? So it's literally metal in there. Or like hemoglobin, for example, right? It uses iron, and that's the thing that carries oxygen. So that would be a cofactor. Coenzymes, on the other hand, are organic molecules. So they're, all this kind of stuff that we studied before. So they're carbon and hydrogen based. So again, cofactors, inorganic ions, coenzymes, organic molecules. As far as I'm concerned, that's basically the only thing you need to know about cofactors and coenzymes. Well, that at the end of the day they help enzymes do their jobs. First of all, let me check this, make sure everything's good online. Sorry, guys. All right. No questions online? Okay, just check. Anyway, your book also points out that both cofactors and coenzymes participate in reactions without being altered. So the recycled and reused, which makes sense because, again, enzymes themselves are not used up, right? They're not changed. So that kind of makes sense. And then finally, the last thing your book points out is that vitamins often serve as a source of coenzymes. So you could write a whole paper on that if you want for independent work. Like what vitamins? Your book mentioned vitamin C, and there's some other ones, but you can look that up. Like which vitamins act as coenzymes, and which enzymes do they work on? I'm pretty sure it would be pretty specific. Anyway, any questions about this slide? All right, so while we're talking about inhibition, or I guess we'll go back to talking about inhibition, there's a different type that you should know about, and it's called feedback inhibition. And this is simply using the product of whatever it is, whatever reaction that you're making, that product itself will turn it into an inhibitor, which makes sense because, again, if your cell needs whatever, the enzyme is making whatever, if it keeps making it, then it makes too much of it, you need to stop, right? Because eventually you'll need it anymore. So what happens is if there's a lot of product, then that product will end up being the actual inhibitor for its own enzyme. So pretty good way of working it, I think. And again, that's just an example. You don't need to memorize that picture or anything. Again, the point is this enzyme is producing a product, and once it's made enough of it, that product itself will be inhibited. Any questions about feedback inhibition? All right. Say the next word for attendance will be square. This square right here. Now we can jump into the other part. So basically what we just talked about is on the first study guide I shared with you guys yesterday. What we're about to talk about is on the next study. Glycolysis. We're going to actually talk about, now we're finally jumping into cellular respiration. So here are the learning objectives. By the end of this section, you'll be able to explain how ATP is used by the cell as an energy source and describe the overall results in terms of molecules produced off the breakdown of glucose by glycolysis. Here we go. First, let's talk about ATP itself. Again, this is all about energy, right? Cells need energy. Cells need activation energy to make these chemical reactions happen. Because without those chemical reactions, we wouldn't be alive. So where does that activation energy come from? Now we know it's lowered by enzymes, but where does that energy come from? As far as we're concerned, it's always ATP that does the trick. You can look this up for independent work if you want. There are other molecules that are used occasionally, but ATP is the big one. Well, most important one, so that's the one we focus on. It is a small, simple molecule. You don't need to know that. It does contain potential energy for cellular work. That kind of goes into what we're saying anyway, right? This is the ATP is the energy. It's almost like a rechargeable battery, basically. It gives these cells the energy they need to do their job. Just like a rechargeable battery, it loses its energy because it's giving up its energy to other processes. And when it does that, it can be recharged. And that is what cellular respiration is all about. It's basically recharging those rechargeable batteries. Cellular respiration breaks down the food you eat. That energy is released. Then your body takes advantage. Your cells take that energy to recharge the battery, so to speak. Any questions about this side? Very important. ATP is the whole thing's about ATP. This whole exam is almost all about ATP. Point is, cells need ATP. In this chapter, you're going to learn how we make ATP, breaking down food, primarily glucose. And then we're going to kind of back it up in the next chapter. Like, all right, well, where did all that energy come from in the glucose that we use to make the ATP? And that's what photosynthesis is. But it's all about ATP. That's the end of the story, right? Trying to get to the ATP. Here's a picture. This is actually from your textbook. This picture. You don't need to know all the parts. And as a matter of fact, I don't even like that picture. It doesn't really tell the story that I want to tell. So here's the picture from a different textbook. And again, now that you need to know the parts. But remember what I said. However much energy is used to create a molecule, that's how much energy, for the most part, is available to be released. So obviously, if we're talking about ATP, it's like the rechargeable battery. We need a really energetic molecule. So not that you need to know the chemistry of this, but look, there's a negative, 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 negative. Another thing you don't know, probably because we're not a consistent chemistry class, oxygens themselves are very negative. They have a lot of electrons. So even though that's not fully negative, there's a lot of electrons there. There's a lot of electrons in double bonds. The point is, these things are really negative. And remember, negative things repel each other. So those little three balls do not want to be together, right? They want to repel each other. So it's going to take a lot of energy to squeeze that third ball onto the second. And again, however much energy it took to build that molecule, that's how much energy is going to be released in the Raik's test. That being said, let's look at this really quickly. Yeah, it's an adenosine molecule. It has a tail of 3 phosphate groups, like we already talked about. When it's broken down, when it's used, and this next picture I'm going to show you is better, but we just lob off that phosphate group, right? So then ATP turns into ADP. So ATP is the fully charged battery. ADP is the dead battery. So again, that should be a test question. I think it will be. How does this work? How does ATP work? It transfers a phosphate group, right? It gets rid of a phosphate group. It loses a phosphate group, however you want to say it. When it does that, that releases a lot of energy. And then your cells can use that energy to do whatever work they need to do. For example, in the last chapter, we were talking about not facilitated diffusion, but active transport, right? So sometimes your cells need to bring things into the cell, even though there's already a high concentration. So that required energy. And I said that energy came from ATP. That's how it works. The next slide I'm going to show you is the visualization of the words that are on the slide. Any questions about this? All right. Again, this picture is not from your textbook. It's a good picture, I think. So there you go. You got that triphosphate, that fully charged battery. That's ATP. Those balls do not want to be together. They're ready to go away. And once one of them does, that releases a lot of energy. So then you have your dead battery, your ADP, plus that extra phosphate. This has been transferred to another molecule. But again, to me, that's the less important part. It doesn't matter where it went. The point is, you had ATP. You got rid of the phosphate group, and that released a lot of energy. So, any questions about that? And then the same thing on this side. Again, the ATP energizes the other molecules by transferring the phosphate groups to them. That's what your book says. And I don't even think we need to focus that in that deep. The point is, you lose that phosphate group, you release energy. Here's some other pictures, not from your textbook. This is from the other textbook. Just so, to move your arms or your muscles, right? You're going to need ATP. And it kind of shows how that works. The ATP transfers a phosphate group to the muscle and it moves. Like we were talking about earlier, bring solutes in. So active transport. You need energy to do it. Transfer a phosphate group. There's your energy, and you can pump those things in. Now we're building something, right? So we're trying to build a molecule. We're trying to build an XY molecule. We have an X, we have a Y. ATP transfers a phosphate group. There's your energy to squeeze those things together, and then you have the product of your reaction. Again, those are just examples. You don't need to know that. What your book does not tell you, so if you take notes, write this down because it's not in your textbook. I want you to know it, and I'm not sure if I'm going to ask it on the exam or not yet, but cells spin ATP continuously. You don't necessarily need to know that sentence because what you do need to know is this right here. There's about 10 million ATPs consumed every second in a single working muscle cell. Of course, I'm not going to ask you, is it a muscle cell or a kidney cell, whatever. The point here that I think what you need to know is about 10 million per second is what you need to know. That's how many cells are being used. Yes, technically it's a working muscle cell. And if you want to, for independent work, you can look up other stuff. What about a non-working muscle cell? What about a kidney cell? What about a skin cell? What about a neuron? How many ATPs you use up? And then you could also do some math. That's just for one cell per second. So you have to wonder how many ATPs are you using in your entire body per second, or per minute, or per day, right? Big numbers. Another thing your book doesn't talk about, and I have it on this picture right here, but it's important. I mean, it's a very important concept. This is what this whole thing is about, is the ATP cycle. I basically talked about it already, but now you can see it visualized. You have ATP, you got your fully charged battery, it releases the phosphate, which releases energy, and now you have your dead battery, the ADP. So then we need a source of energy to squish that third phosphate back on. That's what we have for chemical respiration. And then we have the full battery again, and the cycle continues. It's the easiest cycle there is in biology, as far as we're concerned. It's a full charge battery and then dead battery, basically, right? It's the 3 phosphates or 2 phosphates. Anyway, any questions about this slide? So remember, that's not on your textbook, so you're taking notes. Make sure you have that written down. Make a mental note that you need that to study your slides too, not just your textbook. Here's another picture of what I'm trying to show you. It's from the other textbook. So again, you have your fully charged ATP. You release a phosphate. It releases energy. Now the opposite needs to happen. You need an input of energy to put the phosphate back on, and then you have ATP. And the cycle continues. Any questions about that? Okay. Now we're getting into the meat and potatoes. Here we go. This is the really important stuff here. This is what you need to know for a majority of the exam. It's the 3 stages of respiration. So if you're studying, I mean, everyone's different, but my recommendation is first, memorize the 3 stages. And know that that's the order that they go. First, it's glycolysis. Then it's citric acid cycle, aka Krebs cycle. I'm only writing that. You do not need to know that it's called the Krebs cycle. I'm only telling you that because if you study on your own, if you're reading some other source or watching that video that I shared with you, if you hear Krebs cycle, you know, what the heck? He never said Krebs cycle. The textbook never said Krebs cycle. Just so you know, those are the same thing. I like to call it the citric acid cycle anyway, and you'll see why later. So it's a hint. Citric acid cycle will help you get the correct answers on the exam. But anyway, then there's the third part, which is the electron transport, which is what I usually call it. And I think that's what they call it on that video that I shared you. But your textbook calls it oxidative phosphorylation. So yes, those are same as far as we're concerned. So again, my recommendation is first memorize those 3 things, right? Memorize and and then you can start adding details. For example, what are the products of glycolysis? So I'm going to teach you. What are the reactants in glycolysis? What are the reactants in the citric acid cycle? What are the products of the citric acid cycle? The reactants in the electron transport, the products. Which of those 3 stages produces carbon dioxide? Which of those 3 stages uses oxygen? Which of those 3 stages produces water? Which of those 3 stages uses glucose? These are all things you're going to need to know. So keep that in the back of your mind as we go through this. Another thing your textbook doesn't do, but I'm going to do because your old textbook does it, First, we're going to do a really broad overview of these things. I'm going to go step by step and tell you the big stuff. And then once we have the big overview, then we'll go back and talk about it in detail. If you download the PowerPoint, you can watch this little video that shows it in action. But here you go. I'm going to go through this quickly because this is only the overview. We're going to come back and go through detail. But the first step is glycolysis, and that is when glucose is split into two molecules of pyruvate. And this almost always happens in the cytoplasm. So again, I'm going to give you all this information again later. These are the test questions right here. Which of the three stages uses glucose? Glycolysis. Which of the three stages produces pyruvate? Glycolysis. Which of the three stages happens in the cytoplasm? Or in other words, not inside of the mitochondria? Glycolysis. So any questions about that? Again, this is just the overview. That's the big picture. We're going to go back and talk about that in detail. But the overview is important because there's a lot of test questions there. All right, the next one, citric acid cycle. Again, aka Krebs cycle, depending on if you're listening to this somewhere else. This is the second cycle, or the second stage. This happens within the mitochondria. So again, another test question. Which of the three stages happens inside the mitochondria? Well, this is one of them. The other one also has electron transport chain also happens in the mitochondria, because yes, citric acid cycle does. And it completes the breakdown of glucose down to carbon dioxide, which then releases a byproduct. So again, test question. Which of the three stages produces carbon dioxide? It's citric acid cycle. Now I can give you a little hint that I was talking about. One of the reasons I love to call it the citric acid cycle instead of the Krebs cycle is because all the C's. You can remember, citric acid cycle produces carbon dioxide. It's all a bunch of C's, right? Likewise, as you're going to learn in the next chapter, when we talk about photosynthesis, there's also another cycle, but it uses carbon dioxide. So if you can just remember, anytime we're talking about a cycle, you should think carbon dioxide. There's a cycle in respiration that produces carbon dioxide, and then there's a cycle you're going to learn about later in photosynthesis that uses carbon dioxide. But whenever there's a question about carbon dioxide on this exam, the answer is going to be some sort of cycle. Hopefully all those C's will help you remember that. All right. So again, the whole purpose of ATP is the whole purpose of cellular respiration is to produce ATP, right? So it is worth noting that glycolysis and citric acid cycle do generate a small amount of ATP. That will be a test question too. Which of the three stages produces ATP? They all do. And your book gets even specific. I think your book gets specific and tells you how many glycolysis produces and how many citric acid produces. I don't care about the number. You don't need to know that. You need to know that those two produce a little bit. Here's what I haven't told you yet. Big story here is the electron transport chain, the last stage. It's all about electrons. This last stage is where we make most ATP. So basically, the first two stages, their whole purpose, they do produce a little bit of ATP, but the whole idea here, it's the first time I'm telling you this, it's an important concept. Your book doesn't talk about it too much, or they don't emphasize it as much. The idea here is to strip away high energy electrons. So that glucose has high energy electrons in it. So we break it down into pyruvate, strip away high energy electrons. And then we break that pyruvate down to carbon dioxide. We're stripping away high energy electrons. Because in the third stage, we're going to use those high energy electrons. And we'll talk about how later. But we need high energy electrons for that third stage to produce all those ATPs. So again, this whole idea of cellular respiration, it's all about stripping away high energy electrons because we're going to use those to make ATP. Here's where it gets a little bit tricky, but not too much. When we're stripping away these high energy electrons, there are molecules that move them basically from place to place. That's the molecule that moves it. NAD plus is the thing that accepts the electrons. But technically, once it has the electrons, it's technically a different molecule, and it's called NADH. At any time you see any molecule that looks like this, and there's some other ones that are slightly different, but also they just look the same. Like it looks, FADH2 is the other one. We'll talk about it later. Then there's one in photosynthesis that it looks like this. So when you see anything like this on the exam or the study guide or whatever, you should think, oh yeah, that's the thing that's moving the electrons from one place to the other. And technically, and I'm probably not going to get this detailed, but if it helps you remember, again, the NAD plus is the one that's ready to accept the electrons. I think the easy way to remember that is because it has a positive, right? Obviously, that thing's positive. It's going to be attracting electrons. So hopefully that can help you remember. This is the thing carrying the electrons. It just hasn't gotten its electrons yet. Hence, it's a plus. And then this is the thing that actually has the electrons. So again, and I'm not going to try to trick you. Whenever you see any word looks like this, or even that, you should think, all right, that's a thing that's moving electrons from place to place. So any questions about this slide before we talk a little bit more about the electron transport chain? Remember, this is still the overview. We're still going to go back and talk about these in detail. All right, electron transport chain. So again, like I said, the whole purpose of those other two things was just to strip away high energy electrons. So now finally, the electron transport chain, they're going to be used. Or as the book puts it, electrons are stripped of energy a little bit at a time until finally they're combined with oxygen to form water. So we'll talk about why once we talk about this in detail, but that is another test question. Which of the three stages uses oxygen? And the answer is the electron transport chain. Which of the three stages forms water? And the answer is electron transport chain. And another test question you're going to have that's on the second study guide that I gave you is it's going to be questions about the electrons. So the electrons were stripped away, right, from the glucose. Then they went to the electron transport chain where they were finally accepted by oxygen. And once that happened, that made a molecule of water. It's very important to know that. Oxygen accepts those electrons and that forms water. And if you're going to learn that, that's going to help you with photosynthesis too, because the opposite happens with photosynthesis. In photosynthesis, we're trying to put high energy, we're trying to build glucose, right? So we need high energy electrons to build into that glucose. And guess where the electrons come from? They come from the water. So the water gives up the electrons and that produces oxygen, right? So it's completely opposite. Here, the electrons are accepted by oxygen, which forms water. That will be a test question. All right, so the electron transport chains, proteins. I'm going to show you again. We're going to go into detail later. I'll show you what it looks like. We'll think a lot more since then. They are embedded within the inner membrane of the mitochondria. I'm not going to ask you that, but it will help you remember how it works. So remember, the mitochondria has an outer membrane, just like our cells have a membrane. But then there's also an inner membrane. It doesn't look like this. It's actually a lot more curvy and wrinkly for a reason. And we'll talk about that later. But yeah, there's little proteins in here. This is all where the magic happens. All these little proteins embedded in the inner membrane. And that will be important later. Not for a test question, but just to understand how it works. You need to understand that there's an outer membrane and an inner membrane. Yeah. And again, we're going to talk about this in more detail later. This is still just a big overview. So the overall equation of respiration shows that glucose and oxygen are rearranged to form carbon dioxide and water, right? Glucose plus oxygen gives us carbon dioxide and water. And more importantly, we're making a bunch of ATP. And I love, so the old textbook says 32. A lot of textbooks say 38. That video I shared with you says 38. So for the test, just think, you can memorize either one, 32 or 38. I'm not going to try to trick you. That would be really horrible. And he's like, all right, it's multiple choice exam, and I give you 32 and 38. It's not going to be like that. The choice is going to be like 32, 52, 82, you know. So if you can remember it's in the 30s, then you're okay. But yeah, that would be a test question too. So again, there's a lot of test questions on here. When you study this, you should memorize, you know, glucose is used in glycolysis, oxygen is used in the electron transport chain, carbon dioxide is released in citric acid cycle, water is produced in the electron transport chain when oxygen accepts those electrons. All three stages produce ATP. Anyway, any questions about this overall thing? The next slide is basically another picture of everything I showed you. There it is. So glycolysis, that's the one that happens outside of the mitochondria. It starts with glucose and ends with, let's see, that's the old textbook. Pyruvic acid is the same thing as pyruvate. So we call it pyruvate. And you'll see why. That's another hint I've done, another thing I've done to help you with the exam. You'll see why later. But anyway, again, start with glucose, end with pyruvate. Then that stuff goes into the mitochondria, where citric acid cycle happens, in both of which you're making a little bit of ATP. More importantly, you're stripping away high energy electrons Those high energy electrons are carried to the electron transport chain where they release energy, and that release of energy is used to make a lot of ATP. So again, a lot of test questions here. Which of the three stages happens outside of the mitochondria? Glycolysis. Which of the three stages happens inside of the mitochondria? Citric acid cycle and electron transport chain. Which of the three stages strips away high energy electrons? Glycolysis and citric acid cycle. Which of the three stages uses high energy electrons? Electron transport chain. These are all test questions. Which of the three stages produces ATP? All three stages. Which of the three stages produces the most? Electron transport chain. A lot of test questions right there. So any questions about that picture? So again, this is still the big picture here. This is actually perfect. This is actually funny. The same exact slide that we ended up erroneously on Wednesday. We are now done with the big picture. So now, truly, we're right on track. So Friday, when we get back, we're going to look at all three stages in detail. Then we should be done with this chapter. So that's it for the day. The last word for attendance will be picture. Picture is the last word for attendance. I'll see you guys in lab. Don't be late. Please.